All right, AMP1 students, we are going to continue talking about enzymes. So we are on the objectives where we're going to talk about how substrate concentration, temperature, and pH will affect the enzymes in the body. And then we'll also talk about some inhibitors that will also affect the functioning of the enzymes as well. All right. So we're going to slide down in the textbook here to our picture here showing substrate concentration, temperature, and pH, and what effect those will each have on the enzyme activity. And so enzyme activity, if it increases, that means that the reaction rate increases, the reactions happen, chemical reactions are catalyzed faster, and we get more products. So when we're talking about substrate concentration, and I'm going to enlarge this a little bit more. So what happens when we get more substrate for a particular enzyme is the reaction rate will increase. So, and that would make sense because we have more substrate, so there's more substrate for the enzyme to form a complex with and catalyze that chemical reaction, and then we get more products. So that will, as the concentration of substrate increases, the rate of that chemical reaction increases. It happens faster, and we also will get more products. Now realize though, we don't have an infinite number of these enzymes in the body. So they can be overwhelmed with substrate. So if they get to the point where every enzyme we have is already bound to a substrate, then we can't increase the reaction rate any further or have any more products because the enzymes are kind of working at their maximum ability or potential at that point. So then you will notice the reaction rate kind of level off after that. But usually as the substrate concentration increases, the reaction rate will increase too. All right, and then when it comes to temperature, our temperature in the body, generally we can get increase in chemical reactions when we raise the temperature. Now remember, in the human body though, we can't raise it too high because that will cause damage to proteins. And remember, enzymes are proteins. But there is a normal, optimal temperature where we have like the highest reaction rate for a particular enzyme. And this is actually a little bit higher than our normal body temperature. So they're doing body temperature in Celsius. So just 37 degrees Celsius is about 98.6 degrees Fahrenheit. So that's what we're looking at. So if we just go a little bit higher than that, kind of in the fever range, that's when our enzymes are going to be working the fastest and producing the most products. So that's our increasing. If you increase temperature, the rate of reaction increases. So now it will increase up to a point, and then if we go beyond that temperature, that is going to cause these bonds to break between the parts of the protein and cause it to denature. So remember, we talked about denaturing in chapter 2. And this is a protein. So the enzyme denatures those little weaker bonds, like hydrogen bonds. They get broken at that higher temperature. So everything's just moving really fast, and they're running into each other, and those bonds are getting broken. So they will break. And then remember, if they don't have, this is the normal shape. So then if they don't have their normal shape with their normally shaped active site where the substrate binds, they can't function. So then reaction rate will drop considerably and essentially stop. So you won't have any chemical reactions being catalyzed when you get above that optimal temperature. And then we do have, you'll notice at cooler temperatures, the protein is going to be, reaction rate's going to be much slower. So everything is just moving slower and the structure is a little more rigid. So lower reaction rate at colder temperatures as well. So one reason why we like to keep our body temperature in this normal range is we have pretty normal shaped enzymes that can function pretty well, catalyze chemical reactions in a timely manner. All right, and then when it comes to pH, if I can move that, there we go. When it comes to pH, so here's our neutral pH of 7. And so remember, if we go less than 7 pH, remember this is when the hydrogen ions increase, because pH is essentially measuring the hydrogen ion concentration. So if the hydrogen ion concentration increases, then the pH will decrease. And so we call anything less than 7 acidic. And then if the hydrogen ion concentration decreases, then the pH will increase, and we call that basic or alkaline. All right, so you'll notice that in our bodies, like our normal pH, it's actually 7.4. They have like a range here, 6 to 8. But like our blood, the normal pH of our blood and what our body keeps it at all times through homeostasis is 7.4. So that is the optimum pH for all of our enzymes in our body. And then you'll notice if it gets any lower than 7, so if it starts to get a little bit more acidic, or if it starts to get a little bit higher, get a little bit more basic, both of those things will cause these proteins, which these enzymes that are chemically proteins, to lose their shape because they will denature. And then they can't function properly. And if it gets really, really out of whack, the pH, then definitely getting into where chemical reactions aren't happening, and death's probably going to ensue pretty quickly. So, and this pH, like where we keep it in that normal range, it's pretty narrow range. So, it doesn't take much for the enzymes to stop working. Even if you drop down to 7.3, it's still pretty significant. So, or get up to 7.5. All right. Okay, so those are our factors that will influence enzymes. So the last thing we'll talk about is competitive and non-competitive inhibitors. And so sometimes we will have, here's a nice little picture, we can have so in this picture we have the substrate and enzyme. So here's our substrate and enzyme, and again they have that proper shape, complementary shape, so they can bind together and we can get a chemical reaction catalyzed. But we can sometimes have something called a competitive inhibitor of an enzyme. And a competitive inhibitor is an inhibitor that is going to compete for the active site. So that's why they call it competitive, because it's competing against the substrate for this active site. So you'll notice that it has kind of like a similar shape. So that's why it can inhibit this enzyme, is because the enzyme will like, oh, you have the right shape, so you can bind here to me. But the problem is when the competitive inhibitor binds to this enzyme, the enzyme can't do anything. So it's not the right substrate, so it doesn't catalyze a chemical reaction. So, so it interferes with that normal functioning of the enzyme. And so sometimes this can happen normally in the body. So like sometimes there's body processes where we don't want something to happen at this moment. So we have like natural competitive inhibitors that inhibit some enzymes sometimes, and then they'll go away and let the enzymes function. So it's a way to regulate various processes in the body. But we will have a prescription medication that can serve as a competitive inhibitor. So that's another example of something that can be a competitive inhibitor. So we can use certain competitive inhibitors as a drug, and they can inhibit certain enzymes so that we can kind of regulate certain body processes, either inhibit them or allow them to happen, depending on which enzymes we're going to inhibit. So that's competitive inhibition. And then the last type of inhibitor is a non-competitive inhibitor. And why they call it non-competitive is that it doesn't compete with the active site. What it does is it has a particular shape that will bind to another area on the enzyme. They call it the allosteric site. So there's this little site here is an allosteric site. And so, but what happens when that non-competitive inhibitor binds to the allosteric site, so this little green guy is the non-competitive inhibitor, then what we'll get is a change in the shape of the active site. And so now this enzyme has been inhibited because now this active site isn't the right shape and that substrate can't bind there. And so then it won't be able to work properly. So sometimes that's also a type of inhibitor in the body naturally as well. So some of our inhibitors of enzymes in the body could be non-competitive, cause non-competitive inhibition or competitive inhibition, depending on the particular enzymes in the body. So, all right, so those are our inhibitors that can affect our enzymes. So those are ones that we may talk about in a little bit later in A&P2. Okay, and then the I don't have a number on this one, but I want you to know about this phenylketonuria disease, PKU. So we will talk about that in class. We're going to do a case study on that. Okay, and then the last bit of information from this chapter that we should be familiar with is it's called cellular respiration. And so this is, it goes in the textbook, it goes into a lot of details about cellular respiration. You do not have to know all those details. So what I did with this objective is I put down exactly here in our learning objectives what you need to know about cellular respiration. So realize this is chemical reactions. Cellular respiration is like kind of a combination of chemical reactions that happen in a certain order. And what those chemical reactions are doing are taking organic macromolecules like glucose, which remember is a carbohydrate, fatty acids, those are the components of lipids, and then even amino acids, but not as often. But this cellular respiration this chemical reaction pathway will take those and break them down, and enzymes are involved in that whole process of breaking those bonds between like glucose and the fatty acids. And then each time that bond is broken, you will get that chemical energy that was in that bond, you'll get that released from those organic molecules. And then what will happen is that little energy that's released, the cell, especially if it doesn't need it right then, which sometimes it doesn't, then it will store it in another molecule called ATP, which we talked about in chapter 2, adenosine triphosphate. And so it took the energy that was released when the bonds were broken in glucose and fatty acids, and then converted it into the bond in the ATP molecule. And then the ATP is stored in the cell, and then whenever the cell needs some quick energy, then the cell can break down that bond in ATP and get some energy so that's that chemical or potential energy stored in that bond, convert it to like mechanical energy at that point. So, all right, and the reason that we have this, they call it a metabolic pathway, but when you see metabolic, think chemical reactions, because metabolism just means the sum of all chemical reactions in the body. So these whole chemical reactions that are breaking bonds in glucose and fatty acids and then releasing energy, we have to do it in a stepwise fashion, because remember that second law of thermodynamics, we have that release of heat as well. So if we were to like break every bond in glucose at the same time, we'd get a bunch of energy, but we'd also get a bunch of heat, and that would be too hot for the cell and it would just kind of melt. So we have to do it in a systematic, slow, multi-step process, so that way, and then once we get that energy released, then the cell is stored in ATP for later. So, all right, and then these are the steps that you should know that go on in cellular respiration, but don't worry about memorizing all the numbers of these molecules. So if you have any questions on that, let me know.